Hello everyone, welcome to MES e-learning channel. So this is the continuation of the genome editing series of lecture. In the previous lecture, we had seen the basics of genome editing and what are the mechanisms for the double-stranded repair. And in this particular lecture, we would be seeing one of the mechanisms that we can use for genome editing, that is zinc finger nucleases. So these are engineered proteins. So these engineered proteins are combination of two things. One is the DNA binding domain, which will specifically bind to the targeted sequence. And the next one is going to be the nuclease, that is the DNA cleaving domain. So they allow, this entire protein will allow precision targeted modification of the DNA sequences so that we can use it for different purposes. So let us first understand what is zinc finger motif. So this zinc finger motifs, they have a 3D structure. It's a protein and it is characterized. Why it is called a zinc finger? Because it is having an zinc ion which holds the structure together, and it is very important to recognize a particular DNA sequence. So it is held in place, this particular zinc ion would be held in place by 2 cysteine and 2 histidine R groups, which is there within this particular structure, and it is present in tandem repeats. So these repeats will help us in identifying the precise sequence where it can interact. So the zinc finger will hold this particular molecule together, and every motif will consist of 30 amino acids, and it's a combination of your beta and alpha structures together. So how can these zinc finger motifs bring about specificity in binding to a particular DNA? So the DNA binding domain can be engineered in such a way. So you can see that there are different types of ZNFs that are available. So you have zinc finger 1. So ZF1. So it is specifically having a sequence of AAT so that it can bind to your TTA. Similarly, other zinc fingers will have other specificity. So whatever my sequence is there, depending upon that, I will have to choose specific type of zinc fingers and make a combination of that. Combinations will help me, so I can have different combinations, and every combination will be specific for a particular sequence. So this combination and combinations will make this entire DNA binding domain very, very precise and specific for finding a particular DNA molecule. So now that we have understood what are these zinc fingers, let us understand what is zinc finger nuclease. So it's a combination of these zinc fingers with the DNA cleaving fork1 nuclease. So this fork1 nuclease is taken from bacterial sources. So together what they do is, this zinc fingers will help in recognizing the precise sequence where the double-stranded cut is expected. Okay, so it will recognize and bind to this DNA sequences, and then fork1 domain will create this double-stranded break. So now once your double-stranded breaks have been done, what will happen? Now DNA repair has to be done. So DNA repair will be done either by your homology directed repair. So homology directed repair, when it would be done? Only when the target sequence have been already given with this ZFNs. And if there is no such kind of target sequences being provided, then it will go for the end joining, that is your non-homologous end joining, and some kind of your mutations could be created with the help of either insertions or deletions. So this is how your ZFNs are going to work. As I have already explained, DNA will bind, it will cleave and create the double-stranded breaks, and then followed by the repair mechanisms. Okay, so repair mechanisms could be either by NHEJ or your homology directed repair. So if you want to correct it, correct it or you want to repair it, we will have to go for the homology directed, where a particular exogenous gene has to be given as the target gene. So advantages, precision, very important. These your zinc finger nucleases allow specifically targeting desired DNA sequences and will allow the precise genome modification. They are very efficient, high rate of gene editing, and they are very powerful tools. Versatility, as we have already seen that how your different your Z fingers can help us bind to different types of your sequences. It makes a wide range of DNA sequences could be targeted by the help of your ZFNs and customization. So with the help of different types of your zinc fingers, we can customize and tailor our requirements. Depending upon our requirements, it can be customized accordingly. But yes, there could be some limitations also. So one of the most important challenge that we face is the off-target effects. So where we are not expecting, there could be some kind of your unintended modifications and which could really raise the safety concerns, and that's the reason why ethical concerns arise over here. And then delivery challenges. So all these things are fine, but finally we have to deliver these nucleases into the target cells, and this delivering could be a problem. And one more problem is that if we are using these kind of your ZNFs in case of your eukaryotic organisms, so there could be an immune response elicitation because your FOC1 are nothing but your exogenous proteins. So that could create an immune response, and that could limit the therapeutic applications. So this is all about zinc finger nucleases and how it can be used for the genome editing. I hope this small concept is clear in this particular lecture. Thank you.